everyone, welcome back to Biophilia, your all-time study mate. Today we are going to state about the biomolecules chapter. So if you like this video, then don't forget to like, share and subscribe to our amazing channel Biophilia. So about the biomolecules, we have to discuss in detail about four biomolecules that comes as carbohydrates, second will be about proteins, third is lipids and fourth will be nucleic acids. So let us see one by one in detail. Before understanding how the carbohydrates or the lipids or proteins are estimated, we should know that each and every living organism, yani tissue hai, organ hai, is made up of biomolecules. So how are they estimated or how do you understand how much percentage it is made up of? So it is the chemical composition that will help you to understand it. So how will you understand the chemical composition? So take any living tissue, say it be plant origin or animal origin. So take a piece of vegetable or you can take a piece of liver, that is the meat part, and you can grind the tissue with a mortar and pestle and make it into a slurry. Now this slurry you have to add the trichloroacetic acid, which will help to digest the proteins and the other components properly, and then you have to strain the slurry. In the slurry, when you are straining, you will get two components. First component will be the acid insoluble part, which will be at the top of the filter paper or the muslin cloth that is used to strain, and the part which gets filtered out जो beaker में आ जाएगा, that is acid soluble. Why is it acid soluble or acid insoluble? Because you are adding the trichloroacetic acid that is going to give you the acid solubility or the acid insolubility. So now what to do about this part जो ऊपर बच गया है और जो नीचे बीकर में आ गया है. Now what we wanted to do, we wanted to estimate the biomolecules, but what we are getting is acid insoluble and acid soluble pool. But what to do about these? So here we should know that each and every component of the human body, be it like we said that human body is made up of from the soil, and after that it get mixed into the soil. So why is it called so? Because the earth's crust is also made up of some elements like hydrogen, carbon, oxygen, when even the human body is made up of the same elements but in different percentage or different proportions. So all these will together come and form the components like water or proteins or carbohydrates. Now carbohydrates, there are elements like carbon, hydrogen, oxygen and nitrogen. So these elements when come together in specific proportions, they will form the component of the human or the living organism. So biomolecules, when we need to classify them, we say that there are two types based on the size. जैसे एक बड़ा molecule है, एक छोटा molecule है. The smaller molecules are going to be called as the micromolecules. These micromolecules, since these are smaller in size, are able to get filtered out from the cheesecloth or the muslin cloth that we used. So they come into the beaker as the acid soluble fraction. Whereas the molecules which are bigger in size, or they are made up of multiple units of small quantity, these are macromolecules, and since of their size is big, they cannot get filtered out, so they stay in the acid insoluble fraction which is seen above the muslin cloth. So अभी छोटे छोटे molecules कौन से होते हैं? The ones which are monomers. इसको और छोटा नहीं तोड़ा जा सकता है. These kinds of are nothing but the amino acids, simple sugars that come under the carbs, and nucleotides. Now when amino acid is a single unit. When amino acids many come together, they will form proteins. So amino acids को ही बड़े से beads की तरह जोड़ के proteins बनेंगे. So these become macromolecules. Similarly, when simple sugars, say for example it is a lime, when a simple sugar will combine, it will form a polysaccharide, a chain of it. न्यूक्लियोटाइड के लिए भी सेम रहेगा. न्यूक्लियोटाइड भी बहुत सारे जब जुड़ जाएंगे एक के साथ एक, like when you make a necklace out of many beads. Similarly, a single bead is a micromolecule, and when you make a necklace out of it, it will be a macromolecule. Now, what about these macro and macro? We had seen that we have four components to see: carbohydrates, proteins, lipids, and nucleic acids. तो यहाँ पे तो हमने देख लिया about the polysaccharides that come under the carbohydrates. Proteins हो गए, nucleic acids रहे. But what about the lipids? Lipids तो दोनों में से एक में भी नहीं है. So let us see about the lipids. Where do they fall into? So acid soluble pool may, as I told you, the particles which are smaller in size will be into the acid soluble pool, making up the micromolecules, which will be having a weight of about 18 to 800 daltons. Whereas you consider the macromolecules, they will be in the acid insoluble pool, जिनका molecular mass होगा which is more than 1000 daltons. Now, lipids, as I told you, they are not strictly macromolecules. Why they are not strictly macromolecules? This point becomes important because macromolecules के definition में we have seen over here that the molecular weight should be more than 1000 daltons. Then you call it a macromolecule. And other component is it should be acid insoluble. But in the case of lipids, it is acid insoluble, but it is not exceeding its weight above 1000 daltons. It is less than 800 daltons. That makes it lipids not to be a strict macromolecule. यानी वो acid insoluble तो है, but it is not fulfilling the category of the molecular weight to be more than 1000 daltons. Now, why does it become and comes into the acid insoluble pool? Because when you are grinding the tissue, lipids कहाँ present होते हैं? Lipids are present in the plasma membrane. So when the tissue is grinded, the cell membrane, the plasma membrane gets broken down. And once it is broken down, the lipid is not able to mix with the water. क्योंकि acid को dilute किस में किया है? Acid को dilute किया है in water. So oil and water as does not get separated, means it cannot get mixed. Similarly, the lipid remains water insoluble, and that is the reason it is acid insoluble fraction, but it is not fulfilling the weight category to be exceeding more than 1000 daltons. Now let us see about the primary and the secondary metabolites. Primary metabolites क्या होते हैं? These are the organic compounds जो directly first basic metabolic processes से आते हैं. For example, photosynthesis से you are getting sugars like a glucose, whereas respiratory say you are releasing carbon dioxide. That is the first primary metabolites. Now these primary metabolites are going to be helpful for the identification of the function and also the physiological processes. Like for example, primary metabolite example may amino acids, sugars, chlorophyll or hemes are the primary metabolites. But the secondary metabolites are the organic compounds which are not seen in primary metabolism. उनका कोई direct function नहीं है in the growth or development, but these are still useful for human welfare. For example, like there are rubber. So plant is forming rubber as a secondary metabolite. It is not utilizing rubber for its growth or development, but human is utilizing that byproduct for its welfare. जैसे रबर से रबर टायर बनाया, या drugs होते हैं. For example, there is you can say here in the table you can see there are different drugs which are used by humans. There are pigments, there are spices. अभी जो spices में you can see the essential oils भी होते हैं, या there are some alkaloids. All these are actually the secondary metabolites जिसका directly plant को कोई benefit नहीं है, but humans have identified and it is being used for human welfare. Now we have to see in detail about the carbohydrates. Carbohydrates, may this is a summary chart that you can understand that we need to see in detail about the first part, the very basic unit of carbohydrate, that is monosaccharides. So monosaccharides एकदम छोटी यूनिट है, इससे छोटी यूनिट कुछ और नहीं होगी. Now from monosaccharides how other kinds of monosaccharides are derived, we will understand that. Again, this will be the smallest unit. Now, oligosaccharides will be a bit longer chain, and a very long chain will be a polysaccharide, right? So let us understand from the basic, the very smallest unit of carbohydrate, that is monosaccharides. So carbohydrates previously used to be defined as the hydrates of carbon. जैसे नाम में है, break करो words को, you will see that it is giving you an idea that it is hydrates of carbon. But what was the problem in the definition? That hydrates of carbon, यानी कार्बन को जब हाइड्रेट करोगे then you used to get carbohydrate. तो इसका फॉर्मूला ऐसे बन जाता था कि it is in the form of CnH2O and it is in the power n. So ऐसे कुछ बनता था. So now not each and every carbohydrate used to fit into this formula. या कभी कभी ऐसे होता था कि non carbohydrates are also getting used by this formula. जैसे acetic acid ले लो, it is to fit into this formula but it is not a carbohydrate. So that was a big problem for the scientists to define carbohydrates. So later on it was understood and they gave a new definition that we need to understand here, that carbohydrates are polyhydroxyaldehydes or ketones or compounds which produce them on hydrolysis. यानी हो सकता है जैसे मोनोसैकेराइड है तो वो डायरेक्टली पॉलीहाइड्रोक्सीएल्डिहाइड या कीटोन होगा, बट इफ यू आर टेकिंग पॉलीसैकेराइड तो बहुत सारे छोटे छोटे यूनिट्स से बना है. सो व्हेन यू विल हाइड्रोलाइज they will form the polyhydroxyaldehyde or polyhydroxy ketones. So that was the new definition that was approved everywhere for carbohydrates. Now carbohydrates में क्या क्या होता है? Even if you want to make it to the elemental stage, then you will find carbon, you will find hydrogen, you will find oxygen into the carbohydrates. Now carbohydrates are also commonly called as saccharides. Now why they are called as saccharides? Because basically these carbohydrates are oftenly sweet in taste. So since they are sweet in taste, you call them sugars. So sugars को लैटिन में बोला जाता है saccharides. So carbohydrates are also as saccharides. So you call it monosaccharide or polysaccharide. So that is the use of it. Now let us see the functions about the carbohydrates. So carbohydrates जो होते हैं, these are the direct source of energy. For example, आपने potato खाया, starch खाया, या you are eating sugar into your milk, or you are consuming coffee, there you are adding sugar. Anywhere, whenever you are consuming carbohydrates, that will be a source of energy. This source of energy is rapidly used by the body. क्योंकि carbohydrates में से energy receive करना is much more easier. It can also be getting converted into different energy sources. For example, like fat में convert कर दिया, या you are converting it into amino acids. So it acts like a precursor for different organic compounds. It can also act in the structural formation of the cell membrane. जैसे glycoproteins या glycolipids होते हैं. So जो term glyco आता है, the glyco is regarding what? It is the carbohydrate part. जैसे carbohydrate ने protein के साथ bond बनाया, so that is glycoproteins. So it is helping in the structural unit. It will also be in the functional part. For example, in the cell growth, addition, fertilization, वहाँ पे भी carbohydrates are. Then the next part comes in about the structural components. For example, in the insects, exoskeleton formation is done by chitin. So chitin is a part of carbohydrate. Cellulose is also a carbohydrate that gives extra structural support to other organisms. या फिर एक storage form of energy में भी carbohydrates can be used. For example, in animals, the storage form is glycogen. So in this forms, various carbohydrates are helping the cell or the body to function. So let us now see about the monosaccharides. Monosaccharides word को अगर break करते हो, monosaccharides. Mono means one, saccharide means sugar. सो इट इज द वेरी सिंपल कार्बोहाइड्रेट, वेरी सिंपल शुगर, जिसको आप और ज्यादा नहीं तोड़ सकते हो into smaller components. If you try to break it, you will directly land up into getting what? Carbon, hydrogen and oxygen. So वो elemental stage हो जाएगी. But the simplest form that you can get into a carbohydrate is nothing but monosaccharides. जनरली इट इज कंपोज्ड ऑफ 3 to 7 कार्बन एटम्स, और उसमें से भी 3 to 7 में से भी नेचर में सबसे ज्यादा अवेलेबल कौन से होते हैं? इट इज द 5 कार्बन एंड द 6 कार्बन. दीज आर द मोस्ट फेमस पर्सन, or you can say the most famous monosaccharides land up into 5 or 6 carbon atoms. 5 कार्बन एटम में भी सबसे ज्यादा फेमस कौन होता है? राइबोज होता है. And in the 6 कार्बन you have glucose, fructose, galactose. So ribose का example, where can you get ribose into natural, where can you find it? So ribose आपको मिल सकता है in RNA. RNA सबको पता है. So RNA का R क्या होता है? It is ribose. So one of the component of the nucleic acid is nothing but going to be ribose sugar. ग्लूकोज, एवरीवन नोज, ग्लूकोज बहुत कॉमन है, बहुत बार सुना होगा. ग्लूकोज कैन बी सीन इन ग्लूकॉन डी. गर्मियों में ग्लूकॉन डी पीते हो, सो ग्लूकॉन डी इज नथिंग बट ग्लूकोज प्रेजेंट होता है. एंड दिस ग्लूकोज इज गोइंग टू गिव यू डायरेक्ट एनर्जी. ये एनर्जी ऐसी है कि ब्रेन के पास जाएगी डायरेक्टली. once you tried, आपको फेंट होना लगता है, so you drink glucon D. Why? Because it will provide you a direct source of energy to your brain. Even in grapes, the amount of glucose is higher as compared to other fruits. But other fruits भी, कुछ fruits मीठे होते हैं. So why they are sweet? Because the presence of fructose. So fructose is present in fruit sugar. It is also referred to as fruit sugar. Or it can also be found in honey. So honey में भी fructose होता है. And the last one is galactose, which is also found in the structural with other, जो मिल्क में मिलेगा. So मिल्क शुगर का एक कंपोनेंट होता है galactose, but not directly galactose is found in that. So next is derived monosaccharides. Derived monosaccharides क्या होते हैं? जो monosaccharides हमने अभी देखे 5 कार्बन का राइबोज एंड 6 कार्बन का we had seen the glucose, galactose and fructose. So इसमें कुछ छोटा सा modification कर दो, so that will lead you to formation of derived monosaccharides. For example, deoxy sugar. अभी 5 कार्बन शुगर थी हमारे पास राइबोज. अब राइबोज में से इफ यू ट्राई टू रिमूव 1 ऑक्सीजन एटम को, इफ यू ट्राई टू रिमूव, सो व्हाट विल यू गेट? यू विल गेट डीऑक्सीराइबोज. एक हमने डी मतलब रिमूव किया. क्या रिमूव किया? ऑक्सीजन का एटम रिमूव किया. किस में से? राइबोज में से. सो डीऑक्सीराइबोज इज अ डिराइव्ड मोनोसैकेराइड. बट वेयर कैन यू फाइंड डीऑक्सीराइबोज? यू कैन फाइंड इट इन डीएनए. जैसे राइबोज को हमने लिया था इनटू आरएनए, डीऑक्सीराइबोज इज सीन इन डीएनए. फिर अगर हमने एक हाइड्रोजन हटा के, इफ यू ट्राई टू ऐड 1 amino group means here you are removing one oxygen, here you are adding 1 amino group. When you are adding amino group, see in the diagrams below, यहाँ पे हमने क्या किया? यहाँ से हमने एक ऑक्सीजन हटा दिया फ्रॉम द सेकेंड कार्बन. यहाँ पे हमने एक अमीनो ग्रुप ऐड कर दिया, सो दैट विल बी ग्लूकोसामाइन. देन शुगर एसिड होते हैं. एक हाइड्रोजन को हटा के यू हैव एडेड द एसिड ग्रुप, दैट इज COOH. COOH ग्रुप जब ऐड हो जाता है, दैट विल गिव यू एसिडिक ग्रुप. सो ग्लूकोरोनिक एसिड और एस्कॉर्बिक एसिड. एस्कॉर्बिक एसिड क्या होता है? विटामिन C. देन शुगर अल्कोहल. अगर हमने अल्कोहल ग्रुप ऐड कर दिया, सो दैट विल बी गिविंग यू मैनिटॉल, दैट इज शुगर अल्कोहल. सो ऐसे कुछ स्मॉल फंक्शनल ग्रुप्स को चेंज करके यू आर गोइंग टू गेट द डिराइव्ड मोनोसैकेराइड्स. तो ग्लूकोसामाइन कहाँ मिलता है? ग्लूकोसामाइन जो होता है, यू विल फाइंड ग्लूकोसामाइन इन द कार्टिलेजेस. कार्टिलेजेस में ग्लूकोसामाइन is present in a large amount. Ascorbic acid, I told you, it is vitamin C. और mannitol जो होता है, it is found in brown algae. ब्राउन एल्गी का जो स्टोरेज फॉर्मेशन है that will be in the form of mannitol. The next we see is oligosaccharides. Oligosaccharides, again, when you try to break the word, oligo means small and saccharide is sugar. So these are small carbohydrates जो 2 to 9 monosaccharides से मिले हुए होते हैं. So monosaccharides, what do they have? Monosaccharides have 3 to how many? 3 to 7 or 3 to 8 carbon atoms होते हैं, whereas when you have 2 to 9 monosaccharides grouped together you get oligosaccharides. So each unit is joined by, अभी हमने माला बनानी है तो माला बनाने के लिए हम बीच में थ्रेड का यूज करते हैं. So that thread acts like a link between each of the beads. But here how are these monosaccharides linked together? So join करने के लिए जो glue जैसा काम करता है that is nothing but glycosidic bond. So this glycosidic bond is formed between the functional group, that is aldehyde or ketone group of one saccharide, and the other monosaccharide ka alcohol group will be involved. So when aldehyde or ketone group from one and alcohol group from the other, when comes together, they will remove one वाटर मॉलिक्यूल. मतलब एक साइड से OH निकलेगा, एक साइड से H निकलेगा, and they will be connected in between by the oxygen. So इनका बॉन्ड कैसे बनेगा? ऐसे बनेगा. This is nothing but the glycosidic bond. So rest of it, the H2O molecule will be removed out. And H2O निकालने का use क्या है? To make it more compact. It is nothing but called as condensation reaction. What are you doing? You are doing condensation reaction. So this condensation reaction means you are combining small molecules into a longer chain by removing one smaller unit of molecule. But generally smaller unit of molecule कौन सा होता है? Water होता है. It can be some other group as well, but normally it is water, or majorly it is water. So these are the oligosaccharides. Examples में कौन कौन से होते हैं? It is sucrose. So sucrose जो होता है, it is formed by glucose and fructose. Then there is lactose, that is made up of galactose and glucose, and maltose, that is made up of 2 units of glucose. So 2 glucose together gives you maltose. Then lactose is galactose plus glucose. अब lactose क्या होता है? Lactose is nothing but milk sugar. Milk जो थोड़ा सा मीठा टेस्ट करता है. Every milk is a bit sweet. Why? Because it has milk sugar, that is lactose. Sucrose जो होता है, it is found in the cane sugar. Sugar cane सबने खाया है. It tastes sweet. Why? Because it has sucrose present in it. So these are specifically having only 2 units of monosaccharides. So इनको specific नाम दिया जाता है, that is these are nothing but disaccharides. What are these? These are disaccharides. Why di? Because these have only 2 monomer units linked together by the glycosidic bond. Next we see is reducing sugars. Reducing sugars यानी क्या होता है? The sugars which can reduce, यानी ऑक्सीडेशन स्टेट जिसका कम करता है. यहाँ पे ऑक्सीडेशन स्टेट कॉपर की कितनी है? 2 है. यहाँ पे कितनी हो गई? +1 हो गई. So ऑक्सीडेशन स्टेट रिड्यूस हो गई. So the carbohydrates or the sugars that can do this function of reducing the oxidation state, that is referred to as reducing property. अब ये प्रॉपर्टी जनरली हर सैकराइड के पास होती है which have free aldehyde और free ketone group. जिनके पास free aldehyde या free ketone group present है, these can be called as reducing sugars. Now which are those? जैसे हमने carbohydrates की बात की. Carbohydrates में we have monosaccharides, we have disaccharides, we have oligosaccharides, we have polysaccharides. बट इनमें से भी कौन से? तो अबाउट मोनोसैकेराइड्स, ऑल मोनोसैकेराइड्स आर गोइंग टू बी रिड्यूसिंग शुगर्स. ऑल डाइसैकेराइड्स आर रिड्यूसिंग शुगर्स except there is one exception, that is sucrose. Sucrose is an exception. सो ओलिगो और पॉली के बारे में हम बात नहीं कर रहे हैं. वी आर स्पीकिंग अबाउट मोनो एंड डाई. सो ओनली सुक्रोज is not going to be a reducing sugar. Now, why is it not a reducing sugar? Because you can see here the structure. If you compare it to lactose का structure, lactose किससे बना होता है? One molecule of the lactose, one molecule of glucose. So यहाँ पे बीच में you can see this, the highlighted part is the glycosidic linkage, and here you can see that O and OH. Here the reducing sugar can form a bond to remove out the oxidation state. But if you compare it to the sucrose, sucrose के पास can you see there is no free hydrogen. यहाँ पे हाइड्रोजन किसके साथ बंधा हुआ है? यहाँ पे हाइड्रोजन ऑक्सीजन के साथ बंधा हुआ है. यहाँ पे हाइड्रोजन is with CH2O. So there is no possible chance that the hydrogen will come out and reduce the copper. So sucrose for that reason cannot be a reducing sugar. Next, we have to see about polysaccharides. Polysaccharides, again try to break the word. What do you mean? Poly means many and saccharides is sugar units. So when many sugar units come together, that will be called as a polysaccharide. अब हमने बोल दिया बहुत सारे यूनिट्स साथ में आ रहे हैं. So we can have two possibilities. सारे यूनिट्स साथ में आ रहे हैं तो हो सकता है कि सारे के सारे यूनिट्स एक जैसे आए, सेम यूनिट्स आए, या फिर हो सकता है कि डिफरेंट यूनिट्स साथ में आए. सो इफ दे आर सेम यूनिट्स यू कॉल इट एज homopolysaccharide, or if they are different units coming together in chains, they will be called as heteropolysaccharides. So homopolysaccharides is only one type of monosaccharide monomer is going to form a chain, whereas heteropolysaccharides, which we'll see in the next slide, will be showing you different monosaccharides are coming together and forming a chain of polysaccharide. So सबसे पहला एग्जाम्पल होता है फॉर होमोपॉलीसैकेराइड, दैट इज ग्लाइकोजन. ग्लाइकोजन, अभी इसका मोनोमर यूनिट क्या होता है? मोनोमर यूनिट इसका हो जाएगा ग्लूकोज. सो बहुत सारे ग्लूकोज व्हेन गेट कनेक्टेड टू ईच अदर बाय अल्फा 1,4 लिंकेज, व्हिच इज ग्लाइकोजन जो होता है उसके पास दो चीजें होती हैं. एक स्ट्रेट ब्रांच होती है जो बहुत लंबे तक जाएगी, एंड देयर इज ऑल्सो ब्रांचिंग. बहुत सी जगहों पे वो ब्रांच कर जाती है. So the part which is horizontal, the black one, it will be linked by alpha 1,4 linkage. जहाँ पे ब्रांचिंग नहीं हुई है, पर ब्रांचिंग जहां जहां पे भी होगी वहां पे कौन सा लिंकेज आ जाएगा? alpha 1,6 linkage. So because of this, the branching and the non-branching, the linkage will change. Alpha 1,4 for the straight, and the branched part will be alpha 1,6 linkage. Similar case is also going to be seen in the case of starch. Starch जो होता है, starch is also having its monomer as glucose molecules, but iodine से you can differentiate between the glycogen and the starch. ग्लाइकोजन आयोडीन के साथ रेड कलर देगा, जबकि स्टार्च जो है वो ब्लू कलर देगा. So by that you can differentiate between glycogen and starch. Another important point regarding glycogen and starch is going to be glycogen जो होता है it is a storage unit as in animals, but starch is the storage form in plants. So that is another important point to remember about glycogen and starch. Next is cellulose. Cellulose जो होता है it is unbranched beta glucose polymer. It is unbranched, to remember. Starch और glycogen दोनों branched होते हैं. And cellulose, as you very well know, where can you find it? You can find it in cell wall. You can find it in paper pulp or cotton fibers. And cellulose will not have any color reaction with iodine. Why? Because it is not helical, उसमें branching नहीं है, so वो iodine के molecule को hold नहीं कर पाएगा. It cannot stain with iodine, so cellulose will remain unstained by, or it will not react with the iodine. अभी एक important हमने डिस्कस किया था पॉइंट, दैट वाज इट इज अ स्टोरेज फॉर्म ऑफ प्लांट्स एंड एनिमल्स. एनिमल्स में स्टोरेज फ्रॉम ग्लाइकोजन है, प्लांट्स में स्टोरेज फ्रॉम स्टार्च है, But did you notice that both the storage forms comes from the polysaccharides? Why polysaccharides are used as storage forms? Because there is one reason कि जो polysaccharides होते हैं, these are easy to store, ये जगह कम लेते हैं, सो स्टोरेज के लिए इट इज वेरी इजी. वाई? बिकॉज एक मोनोमर को अगर हमने ऐसे ही रख दिया तो इट इज हैविंग अ वेरी हाई चांस कि इट विल सराउंड अ लार्जर एरिया ऑफ वाटर अराउंड इट. But if you try to keep a polysaccharide which is clumped together, you are storing many monomers, and comparatively you are also having very less water that is surrounded to it. So actually, how much amount of sugar are you storing here? Only 1 unit. But in polysaccharides, you are storing, say for example, 100 units. So 100 units की जितनी जगह है उतना ही वो एक unit single खाएगा if it is stored in the form of monomers. So as nature has understood this problem, it has converted everything, every monomer into polysaccharides, and now the polysaccharide is used to store. It can be also, why is it easy to store? Because of majorly three reasons. First of all, एक reason होता है कि उसका bulk जो है वो कम हो जाता है. Again, it will be chemically inactive. So chemically उसमें कुछ there is no chemically changes that is occurring if you are changing it to polysaccharide, and even it is osmotically inactive. मतलब if you are storing it in the form of polysaccharides in very large quantities, तो ऐसे नहीं होगा कि osmolarity की वजह से high concentration to low concentration water is getting attracted. So that is not the case with the polysaccharides. So polysaccharides therefore are easy to store uh, possibles because of these three reasons. The next is inulin. Inulin जो है, it is a polymer of fructose. अभी तक जो हमने देखे glycogen, starch and cellulose, these were the monomers of glucose. But now here you will have inulin which is of fructose. अब fructose जो होता है, inulin जब form होता है from fructose, it is not going to get metabolized in the human body. And since it is not metabolized, it will directly, जैसे का वैसा, will be excreted out from the kidney. So that is the reason when you want to do a kidney function test, किडनी फंक्शन टेस्ट जब करनी है कि कितना फिल्ट्रेशन हो रहा है फ्रॉम द किडनी, you will supply the patient with inulin. You know the fixed quantity how much you have supplied, and you will test the urine after some amount of test period to see how much is excreted out. क्योंकि इन्यूलिन डाइजेस्ट नहीं हो रहा है, इट इज नॉट गेटिंग मेटाबोलाइज्ड, जैसा का वैसा बाहर आ रहा है. सो विद दैट यू कैन एस्टिमेट हाउ द किडनी इज functioning. The last monopolysaccharide that you have to see is chitin. So chitin is the second most abundant organic substance, and it is going to have the N-acetyl glucosamine as its basic monomer unit. सो इन्युलिन में फ्रुक्टोज था, काइटिन में क्या है, नैग है, एन एसिटाइल ग्लूकोसामाइन. ग्लूकोसामाइन क्या है, इट इज अ अमीनो शुगर दैट इज डिराइव्ड मोनोसैकेराइड. और काइटिन कहाँ मिलेगा, एज इट्स होमोपॉलीसैकेराइड फॉर्म, it will be formed in the exoskeletons. Next we have to see is heteropolysaccharides, यानी एक से different types of monomers are going to come and form a chain. So यहाँ पे दो examples याद करने हैं, that is peptidoglycan and hyaluronic acid. Peptidoglycan जो होता है उसमें alternation of amino sugar होती है, that is N-acetamine glucosamine and N-acetyl murameric acid. दो amino sugars है, N-acetyl glucosamine that is your NAG और NAM that is N-acetyl murameric acid. इन दोनों के बीच में alternation होता है, and that alternation will form your heteropolysaccharide that is peptidoglycan. Hyaluronic acid जो होता है, it is going to be composed of glucuronic acid and the acetyl glucosamine. So these are your two units जो वापस in certain pattern will be joined together by the glycosidic linkage and form the heteropolysaccharides. Next we see is about amino acids. So amino acids are the basic monomers to form proteins. So amino acids कैसे form होते हैं? How is the structure? Let us understand that in detail. So amino acids are again organic compounds. Organic compounds है मतलब इसमें कार्बन है, हाइड्रोजन है, ऑक्सीजन है, नाइट्रोजन है. So these is a organic compound. अभी amino acid है. यहाँ पे amino acid में दो words है. One is amino that comes from the term of amino group. और दूसरा एक वर्ड होता है एसिड. सो एसिडिक ग्रुप जो होगा जब कार्बन को कंबाइन करेगा दैट विल गिव यू अमीनो एसिड. सो ऑर्गेनिक कंपाउंड जिसके पास एक साइड पे अमीनो ग्रुप है दैट इज NH2 ग्रुप और दूसरे साइड पे कार्बोक्सिलिक ग्रुप है दैट इज COOH attached to some carbon and rest of the valency is fulfilled by hydrogen or a variable group, then in that case it is called as a amino acid. अब ये इतनी बड़ी डेफिनेशन याद रखने से बेहतर है एक डायग्राम फॉर्म में याद कर लो. So that is easier to remember. So how will you remember that? सबसे पहले कार्बन को बीच में रखना है. कार्बन की वैलेंसी कितनी होती है? 4 होती है. So एक साइड पे you have attached amino group that is most important. The second is carboxylic group because amino acid ऐसा नाम है. फिर rest of it can be given to a side chain that is any other group other than hydrogen or it can be provided to the hydrogen to complete the valency. So that forms a structural unit of amino acid. अब अमीनो एसिड जो होता है ये किससे रिजेंबल करता है? कैन यू इमेजिन हाउ टू रिजेंबल अमीनो एसिड टू? अमीनो एसिड रिजेंबल करता है टू अ सिंपलेस्ट ऑर्गेनिक एसिड दैट इज मीथेन. कैन यू सी मीथेन CH4 होता है. You have replaced one hydrogen with amino group. You have replaced another hydrogen with carboxylic group. You can keep one hydrogen or you can replace it to a side chain. So it is substituted methane. Amino acid को आप क्या बोल सकते हो? You can also call it as a substituted methane. So when both the functional groups are on the same side of the carbon atom, that means अभी ये हमने 2D structure बनाया. But the amino acid actually don't occur in this 2D structure. These are 3D structures. So हो सकता है कि one is on top, other group is on the bottom. But when they are on the same side, दोनों ऊपर है या दोनों नीचे है, then in that case they are called as alpha amino acids. So now you should know that basically we have understood about the 20 different types of amino acids. ये डिफरेंट 20 टाइप्स ऑफ अमीनो एसिड्स अलग अलग कॉम्बिनेशंस में आके प्रोटीन्स फॉर्म करेंगे. So these 20 types of amino acids will have differentiation. How will you differentiate into these 20 different types of amino acids? So differentiation आता है because of the R group, that is the variable side chain group. For example, variable side chain group में आपने हाइड्रोजन को हटा के you added methyl group, so that will give you alanine. हाइड्रोजन ही रहने दिया तो ग्लाइसिन बन जाएगा. Instead of methyl group you decided to add hydroxy methyl group, so वो serine बन जाएगा. So likewise once you change the R group, the variations will cause to form different types of amino acids. The next we see is on the classification about the essentiality. Amino acids में essential amino acids होते हैं. That means the humans cannot synthesize, these are incapable to synthesize these amino acids. Body में इनका production नहीं होता है. तो production नहीं होता है, then how can we suffice the need of it? So very important to remember that these essential amino acids needs to be given or needs to be provided by dietary source. मतलब आपको डाइट में से इनको लेना पड़ता है to complete its requirement. So ऐसे कौन कौन से होते हैं amino acids? So that is arginine, histidine, isoleucine, leucine, threonine, lysine, methionine, phenyl, tryptophan and valine. So इन सबको याद करने के लिए you can remember a trick that has any help in learning these little molecules proves truly valuable. ये sentence याद कर लो, you can remember all the essential amino acids that you need to remember. जैसे हर word का starting will be related to the essential amino acids that you need to take by diet, you cannot synthesize by your own inside the body. दूसरा पार्ट होता है दैट इज सेमी एसेंशियल अमीनो एसिड्स. सेमी एसेंशियल है यानी इनका कुछ अमाउंट आपको डाइट से लेना होता है, अदरवाइज बॉडी कैन try to form when it is emergency. So these are arginine and histidine. थर्ड कैटेगरी आती है दैट इज नॉन एसेंशियल अमीनो एसिड्स, जिनको बॉडी बना सकती है, बॉडी कैन सिंथेसाइज एंड दे आर नॉट नीडेड इन द डाइट. So if you are remembering the essential amino acids, तो हमें पता है कि टोटल अमीनो एसिड्स कितने हैं. टोटल अमीनो एसिड होते हैं 20. आउट ऑफ दैट मेजरली व्हाट यू हैव टू रिमेंबर एसेंशियल अमीनो एसिड्स एंड नॉन एसेंशियल अमीनो एसिड्स. अगर ट्रिक से इफ यू आर रिमेंबरिंग एसेंशियल अमीनो एसिड्स, कैन यू रिमूव 20 minus the essential amino acids to remember the non-essential ones? So in that case you can easily rule out कि ये तो essential की list में आता है. So you are say example 4 options दिए हैं, find out which one of the following is non-essential amino acid. आपको पता है B, C और D तो essential है, so essential की category में आते हैं. So for sure A will be coming into the non-essential category. The next we have to see the classification based on the R group. यानी पहले हमने देखा कि उनका कितना essentiality है, हमें उनको डाइट में खाना जरूरी है या नहीं है. So that was one type of classification. Other classification is based on the R group, the changes in the functional groups. For example, अगर हमने एक हाइड्रोजन को हटा के हमने एसिडिक ग्रुप डाल दिया है, so that means that we will be having amino acid which will have one extra carboxylic group, so that will turn into a acidic amino acid, जिसका example होता है glutamic acid और aspartic acid. Second होता है basic amino acid. हाइड्रोजन को हटा के हमने instead of acid group we added one more amino group, so that will give you a more additional basic group. So वो बन जाता है बेसिक अमीनो एसिड, फॉर एग्जांपल लाइक लाइसिन और आर्जिनिन. तीसरा फॉर्म हो सकता है न्यूट्रल, यानी हमने हाइड्रोजन को हटा के ना एक एक्स्ट्रा एसिड ग्रुप डाला, ना हमने उसको एक एडिशनल बेसिक ग्रुप दिया, जैसा था वैसा का वैसे रखा. सो इन दैट केस इट विल बी अ न्यूट्रल अमीनो एसिड, यानी एक अमीनो ग्रुप है, एक कार्बोक्सिलिक ग्रुप है. So 1 plus 1 minus will be equal to 0. So it is neutral. That is in the case of valine, alanine, glycine, leucine और isoleucine. उसमें से भी सबसे बेसिक अमीनो एसिड कौन सा होता है? सबसे बेसिक होगा ग्लाइसिन. ग्लाइसिन का स्ट्रक्चर देखो, here I have showed you ग्लाइसिन का स्ट्रक्चर. One amino group, one carboxylic group and rest 2 are hydrogens. Next we have to see is sulfur containing amino group. So instead of one hydrogen you are replacing it with sulfur. So sulfur जब आप add कर दोगे उसमें, that will give you sulfur containing amino acids, that is cysteine or methionine. Next होता है alcoholic amino acid. Instead of hydrogen you are substituting it with the alcohol group, that is OH group, that is serine or threonine. Next होता है heterocyclic amino group. Instead of one hydrogen you are adding a nitrogen into it, additional single nitrogen in form of a ring. For example, you can see that in the case of proline or histidine. नेक्स्ट होता है एरोमेटिक अमीनो एसिड. अभी तक हमने जो भी देखे, हाउ वर दे? दीज वर हैविंग स्ट्रेट चेन्स, बट नाउ व्हेन यू आर एडिंग अ साइक्लिक स्ट्रक्चर onto a straight side chain, that will give you a aromatic amino acid. For example, in the case of tyrosine, phenylalanine or tryptophan, you can see the aromatic side chain is present. So this is on the different types of R groups that is possessed by the amino acids. Now we have to see the property of amino acid. यानी अमीनो एसिड्स के पास वी हैड सीन दैट इट हैज वन, दैट इज वन अमीनो ग्रुप NH2 ग्रुप है, एक होता है कार्बोक्सिलिक ग्रुप। सो NH2 ग्रुप जो है वो बेसिक है, समझो बेसिक को आपने नेगेटिव चार्ज दिया, एसिडिक है उसको आपने पॉजिटिव चार्ज दिया। सो एक ही कार्बन के ऊपर यू कैन सी दैट एक साइड पे पॉजिटिव चार्ज है, एक साइड पे नेगेटिव चार्ज है। This property of the amino acid to get ionized into either positive or either in negative, that will be called as Zwitter ion, जिसके पास दोनों चार्ज एक ही एटम पे है, that is called as Zwitter ions. So you can see amino acids को अगर हमने acidic ग्रुप में डाल दिया, so it will turn into a acidic or a positively charged. अगर आपने same amino acid को basic मीडियम में डाल दिया, so that will turn into a basic amino acid. So वैसे ही once it can change it ionic property, that will be the property called as Zwitter ion. So अभी हमने एक और चीज देखनी है that is important for the next topic that we are seeing. So दो अमीनो एसिड्स हमने अभी देखा अभी तक कि अमीनो एसिड्स क्या है, it is a single monomer. अब सिंगल मोनोमर को जब जोड़ना होता है तो कार्बोहाइड्रेट्स के केस में व्हाट डिड वी यूज, वी हैड यूज्ड अ बॉन्ड, और वो बॉन्ड का नाम हमने कार्बोहाइड्रेट्स में क्या किया था, ग्लाइकोसाइडिक बॉन्ड. But in the case of two amino acids to be joined together, this joining will be done by peptide bond. कौन सा बॉन्ड फॉर्म करेगा, पेप्टाइड बॉन्ड फॉर्म करेगा. सो व्हेन फ्यू अमीनो एसिड्स आर जॉइंड टुगेदर इट फॉर्म्स अ ओलिगोपेप्टाइड, एंड व्हेन मेनी अमीनो एसिड्स कम टुगेदर इट विल फॉर्म अ पॉलीपेप्टाइड. सो ओलिगो कौन सा है, पॉली कौन सा है, पॉलीपेप्टाइड को ही हम बोलते हैं proteins. That is our next topic. So when amino acids are combining more than 50 in number, then it comes into the category of polypeptide. Otherwise it is a protein. Proteins जो है उनको 50 से ज्यादा होना चाहिए. If they are less than 50, then it is a polypeptide. And if they are very small, say for example 2 to say 15 या 30 amino acids hai, so that is a oligopeptide. Next we see is about proteins. So here in the image you can see proteins are in various forms. So let us see in detail about these different forms of protein. So first of all, what is protein? Protein is a macromolecule. This macromolecule is a large size molecule because it is going to have at least 50 amino acids which are linked together by a peptide bond. So proteins जो हो सकते हैं, it can be a heteropolymer and it is never a homopolymer. जैसे हमने कार्बोहाइड्रेट्स में पॉलीसैकेराइड्स किया, तो पॉलीसैकेराइड्स में we had seen that there can be a homopolysaccharide or a heteropolysaccharide. So here in this case, proteins are never going to be a homopolymer. These are always made up of different amino acids together to form a heteropolymer called as protein. Now, who is going to be the most abundant protein? सबसे ज़्यादा quantity of protein कौन सा होता है? It is an enzyme that is called as ribulose biphosphate carboxylase oxygenase. It is a enzyme which is going to be used in the photosynthesis to convert it into sugar. The hydrogen, the carbon is fixed into form of ग्लूकोज. सो वहाँ पे रुबिस्को का यूज होता है विच इज द मोस्ट अबंडेंट प्रोटीन इन द होल बायोस्फीयर. इंपॉर्टेंट टर्म यहाँ पे कौन सा है, होल बायोस्फीयर है. इफ यू आर आस्क्ड अबाउट द मोस्ट अबंडेंट प्रोटीन इन द एनिमल वर्ल्ड, तो एनिमल वर्ल्ड में कौन सा होगा, इट इज द कोलेजन. बिकॉज रुबिस्को कहाँ मिलता है, रुबिस्को मिलता है इन द प्लांट्स. It is not found in the animals, but still plants की quantity ज़्यादा है. So entirely in the whole biosphere, Rubisco will be the most abundant protein, and specifically to animal world, it will be the collagen. Next we see is the structure of proteins. सबसे पहला basic structure होता है, that is primary structure, which is a sequence. जैसे एक माला बना दी, सीधा हमने क्या किया, अलग अलग कलर के बीड्स लिए और सबको एक डोरी से जोड़ दिया. So that is a primary structure. Simple a polypeptide chain is nothing but a primary structure. इससे आपको क्या पता चलता है? You will get the positioning information. आपको पता चलेगा कि सबसे पहला बीड जो है, that first bead is for example say of black color, उसका राउंड शेप है. सेकेंड बीड जो है वो ग्रीन है. थर्ड बीड जो है वो येलो है. फोर्थ बीड जो है फॉर एग्जाम्पल इट इज ब्लू. अगेन फिफ्थ इज ग्रीन. सिक्स्थ इज ब्लू. आपको ऐसे उसका पोजिशनिंग इंफॉर्मेशन पता चलेगा वंस इट इज इन द प्राइमरी स्ट्रक्चर. सेकेंड यू कैन गेट इज सेकेंडरी स्ट्रक्चर. सेकेंडरी स्ट्रक्चर यानी प्राइमरी से थोड़ा और हमने मॉडिफाई किया, हमने और थोड़ा एडवांस्ड स्ट्रक्चर लाया. सो सेकेंडरी स्ट्रक्चर विल बी शोइंग यू अल्फा हेलिक्स और बीटा प्लीटेड शीट. अल्फा हेलिक्स जो होता है, there will be a interaction, there will be a bond formation, that is hydrogen bonds form हो जाएंगे between every fourth amino acid, and this will give you a alpha helix structure. Alpha helix कौन से proteins में मिल सकता है? You can find it in the keratin of hair. You can see the right helix. Whereas when the two or more polypeptide chains are held together by hydrogen bonds, then you get it a beta pleated sheet. अब यहाँ पे hydrogen bonds alpha के केस में कौन से थे? It was intramolecular. वो एक ही polypeptide chain के अंदर bond form हो रहे थे. But in the beta pleated sheet you can see that it is forming between two polypeptide chains. So example for beta pleated sheet can be the natural self. Natural self में जो प्रोटीन है उसका स्ट्रक्चर जो होगा it is a beta pleated sheet structure. Third type of structure is the tertiary structure. जो tertiary structure होता है it is much more complex structure than the primary or the secondary. Primary में we saw that there was a straight chain. This was primary. Secondary में what did we do? We introduced hydrogen bonds that gave you a helical structure or a beta pleated structure. Tertiary में you are interwoven it. Now this interwoven is forming a hollow woolen ball like structure. It is a 3D view of protein. अभी तक जो पहले के दो थे वो 2D format में थे. From the tertiary structure it is forming a 3D format of protein. And this type of thing can be formed in glue or the enzymes, which is in the form of tertiary structures. Fourth होता है quaternary structures, जो और एक complex form है of the structure of protein, wherein there are more than one subunits or polypeptide chains that are either in the form of primary, secondary or tertiary, और इन सबको combine करके बनाते हैं quaternary structure. Then this quaternary is found in only one part of example, that is hemoglobin. Hemoglobin जो होता है उसके पास 4 helical polypeptide chains होते हैं. One is from the 2 alpha chains and 2 beta chains. तो समझो रेड जो ऊपर डार्क में है वो 2 अल्फा चेन्स हो गए और नीचे वाले हो गए 2 बीटा चेन्स. So this will be giving you a quaternary structure of hemoglobin. Next we see the functions of proteins. Functions of proteins जैसे हमने कार्बोहाइड्रेट्स के functions देखे थे वैसे proteins के क्या क्या functions होते हैं. So proteins are going to be involved in the defense mechanism. डिफेंस यानी अगर समझो कोई बाहर से एंटीजन आ रहा है टू प्रोटेक्ट आवर बॉडी, these proteins are going to help. These are in the form of immunoglobulins or in the form of antibodies जो आते हैं from the B lymphocytes. These can also be chemical messengers in the form of hormones. For example, insulin. Insulin is a protein or peptide hormone. Receptors होते हैं in your nose, ears for sensation. These are also formed by proteins. There are some transport proteins like GLUT4, which is helping in glucose transportation. So that is one example for transport protein. या फिर hemoglobin, which is a quaternary structure protein, which is also helping in the transportation of oxygen. या फिर structural usage भी होता है proteins का in the form of collagen. Collagen is the main protein in the connective tissue that will be having high tensile strength, and it is the most abundant protein in the animal world. So that was about the different 5 functions of proteins. Let us move on to the third type or the third subtopic about biomolecules, that is lipids. लिपिड्स जो होते हैं these are again made up of organic molecules. So elements कौन कौन से होंगे? Carbon, hydrogen and a bit of oxygen. Oxygen content in lipid is comparatively lower than compared to others. Lipids जो होते हैं we very well know these are oil based. These are indirectly oils. So these are water insoluble, but these can get soluble or these can get dissolved in organic solvents like ether, benzene or acetone. These are not polymers, very important to remember. ये polymers नहीं है, but these are going to be involved in the acid insoluble fraction because these are water insoluble. इसकी वजह से वो acid के अंदर mix नहीं हो सकते हैं. So lipids could be as simple fatty acids or they can be glycerol, whereas they can also form complex structures like in the case of phosphorus containing ग्रुप्स हो सकते हैं या ग्लिसरॉल फैटी एसिड्स को कंबाइन करके लिपिड्स फॉर्म हो सकते हैं. सो देयर कैन बी सिंपल एंड अ बिट कॉम्प्लेक्स लिपिड्स एज वेल. सो लिपिड्स, सिंपल लिपिड्स कौन से होते हैं? ट्रू फैट्स या वैक्सेस. देर आर कॉन्जुगेटेड और कंपाउंड लिपिड्स जो एक लिपिड मॉलिक्यूल के साथ किसी और के साथ बॉन्ड बना के दे आर फॉर्मिंग फॉस्फोलिपिड्स, ग्लाइकोलिपिड्स, लिपोलिपिड्स और क्रोमोलिपिड्स. एंड कुछ छोटे मोटे चेंजेस करके लिपिड्स के अंदर यू कैन फॉर्म डिराइव्ड lipids. So this is a small chart that can give you a summary about how the lipids are classified. So first we will see about the simple lipids. Simple lipids जो होते हैं उसमें दो categories आते हैं, the neutral और true fats और second आता है waxes. So neutral या true fats जो होते हैं these are nothing but the esters of fatty acid. These esters of fatty acids will have alcohols in them. For example, glycerol. Glycerol molecules होते हैं which are going to have 3 carbon atoms which will be bearing hydroxy group, that is OH group. So that is glycerol. Second होता है fatty acid molecule जो attach होता है. These are unbranched chains of carbon atoms which are having carboxylic group attached to a R group. For example, palmitic acid जिसमें 16 कार्बन होते हैं. Then there is arachidonic acid which has 20 carbon atoms. So likewise there are different fatty acid molecules. अब ये दोनों जब साथ में आते हैं then you will be forming the neutral or the true fats which are esters of the fatty acid. Next we see is waxes. These waxes are also esters of fatty acids of higher molecular weight. अल्कोहल यूज होता है बट अल्कोहल ट्रू फैट्स में कौन सा होता है? ग्लिसरॉल होता है. बट हियर इट इज इंस्टेड ऑफ ग्लिसरॉल. ग्लिसरॉल को छोड़ के अगर कोई और अल्कोहल यूज हो रहा है देन इट विल बी कमिंग अंडर द कैटेगरी ऑफ वैक्सेस. फॉर एग्जांपल बीज वैक्स होता है. Now beeswax का तो use most of the people females so very well know about the use of beeswax. It is going to be used in different kinds of कॉस्मेटिक प्रोडक्ट्स में तो बीसवैक्स बहुत ज्यादा यूज होता है. लिप बाम में हो सकता है, मॉइस्चराइजर्स में हो सकता है. So it also be a protective role होता है for the waxes. It is also forming a insoluble coating onto the hair, skin and stem and leaves. So naturally भी present होता है and even other animals like wool bearing animals, wool जो बनाते हैं तो sebaceous glands में से जो secretions आते हैं of the inulin, these are nothing but the wax which is called as wool's wax. What do you call inulin as? इन्यूलिन होता है वुल वैक्स. जो वुल शीप होती है वो शीप के ऊपर जो एक ऐसा स्टिकी वैक्स होता है दैट इज नथिंग बट लैनोलिन. नेक्स्ट वी सी इज कंपाउंड और कॉन्जुगेटेड लिपिड्स. फर्स्ट होता है फॉस्फोलिपिड्स. दीज फॉस्फोलिपिड्स आर कंपोज्ड ऑफ ग्लिसरॉल. ग्लिसरॉल यानी अल्कोहल पार्ट हो गया और कैन हैव एनी अदर अल्कोहल एज वेल इंस्टेड ऑफ ग्लिसरॉल. उसमें एक फॉस्फेट ग्रुप होगा. It will have 2 fatty acids and 1 nitrogen containing choline molecule. So these are the components. एक हो गया glycerol, दूसरा हो गया phosphate group, तीसरा two fatty acid chains and one nitrogen containing group. So when these four things combine together, you will form a phospholipid. So here in the schematic diagram, you can see the presence of choline, that is nitrogen containing. Then phosphate group आ गया, दो fatty chains आ गए, बीच में glycerol को रख दिया. This is nothing but the schematic diagram for phospholipids. Next होता है glycolipids. Glycolipids are going to be containing fatty acids, alcohol, sugar. ग्लाइको है, वी हैड सीन इन कार्बोहाइड्रेट एज वेल. ग्लाइको में क्या होता है, ग्लाइको इज द शुगर पार्ट, लिपिड में तो लिपिड है. सो व्हेन लिपिड इज कंबाइनिंग विथ शुगर मॉलिक्यूल, that is nothing but your glycolipid. Yani instead of two fatty acid chain, when one fatty acid chain is replaced by sugar molecule, that is called as glycolipid. Glycolipids कहाँ मिलते हैं, glycolipids मिलते हैं in the myelin sheath of nerve fibers and also as the outer surface for the chloroplast cell membrane. Now third part is lipoproteins. Lipoproteins yani instead of one fatty acid chain, there are going to be lipid and proteins together. That is lipoprotein. Fourth होता है chromolipids. Chromo is the terminology that is used for color. So उसमें pigments होते हैं. For example, carotenoids और lipids साथ में आ गए. That will give you carotene या फिर vitamin A. So beta carotene can get converted into vitamin A in most of the animals. So beta carotene and vitamin A are the lipids that come under the category of compound or conjugate lipids because of chromolipid category. नेक्स्ट आता है अबाउट द डिराइव्ड लिपिड्स. डिराइव्ड लिपिड्स यानी इंस्टेड ऑफ फैटी एसिड्स, these will have different fat like properties, जिसकी वजह से इसको लिपिड्स के अंदर रखा गया है. Lipids के we have seen what are the components. One component should be alcohol, majorly it is glycerol once it is a true or neutral fat. और second component होता है fatty acids. So in derived lipids, say in the case of steroids, they do not have fatty acids, but they behave like they have some fat in them. So because of that, they are kept into the derived lipid category. So steroids जो होते हैं, these are going to have 4 fused carbon rings instead of a straight chain. अभी तक जो भी हमने lipids देखे, these were all in straight chain. But steroid is going to be having 4 fused carbon rings, and most common steroid is nothing but cholesterol. Cholesterol सबने नाम सुना है. Cholesterol is nothing but a lipid, but specifically it is a derived lipid, that is a steroid. So cholesterol जो होता है, it is the most abundant steroid in the animal tissue. Most abundant protein in biosphere कौन सा होता है, it is rubisco. Most abundant protein in animal world is collagen. वैसे most abundant steroid in animal tissue is going to be cholesterol, and cholesterol से ही body can synthesize acetic acid, so it is a precursor for acetic acid formation as well. और कोलेस्ट्रॉल सिंथेसाइज हो सकता है inside the body in the liver. Liver can synthesize cholesterol. So when an animal is fed onto large amounts of fatty food, बहुत ज्यादा oily food कोई अगर खा रहा है, so liver has a tendency to store and convert that oily food into cholesterol. So जब बहुत ज्यादा cholesterol formation होगा into the body, it will get deposited into the arteries, into the capillaries, that will lead to blockage of the arteries and the veins or the capillaries, जिसकी वजह से heart attack आ सकता है, that is atherosclerosis. That is all related to cholesterol. Next we see is nucleic acids. Nucleic acids, we all know the most important nucleic acids कौन से होते हैं. DNA होता है, RNA होता है. DNA और RNA में भी there are two categories, either they are single stranded or they are double stranded. So based on that, let us see in detail about these nucleic acids. Nucleic acids जो होते हैं, these are again polymers, these are macromolecules, that means, और इनका monomer unit क्या है, न्यूक्लियोटाइड है. Now there are two types of nucleic acids, that is DNA and RNA. अब न्यूक्लियोटाइड जो होता है इट सर्व्स एज अ बिल्डिंग ब्लॉक, यानी ये मोनोमर है दैट विल फॉर्म अ न्यूक्लिक एसिड दैट इज डीएनए और आइदर आरएनए. तो डीएनए या आरएनए में व्हाट इज द मोनोमर? इट इज अ न्यूक्लिक एसिड. न्यूक्लियोटाइड के अंदर क्या क्या होता है? न्यूक्लियोटाइड के अंदर यू विल फाइंड थ्री मेजर थिंग्स. फर्स्ट ऑफ ऑल यू विल फाइंड अ फॉस्फेट ग्रुप, you will find a pentose sugar. So pentose sugar we have discussed, there are two types of pentose sugar. Either it can be a ribose or it can be deoxyribose. Suppose if it is ribose, then it is RNA. If it is deoxyribose, then it will be found in DNA. Then there are heterocyclic nitrogen containing compounds which are called as bases. Bases जो होते हैं, these are of two types. One is purine, one is pyrimidine. Okay, so once these are called as purines, these are 9 carbon, whereas pyrimidines are 6 carbon. प्यूरिन में आते हैं ए एंड जी. ए एंड जी क्या है? एडिनाइन एंड ग्वानिन. और pyrimidines में आते हैं तीन, सी यू टी, कट. So C is cysteine, U is uracil and T is thymine. So U जो होता है it is specifically only present in RNA in a replacement to, in किसके जगह पे आता है? In the placement of thymine. Thymine is present only in DNA and RNA में होगा uracil. So that is about the nitrogen containing compounds. You can see the structures of all pyrimidines, purines. You can see the structure of sugar, that is ribose and deoxyribose. You can see the structure of phosphate group, कैसे phosphoric acid से convert होता है into phosphate ion, which is inside a nucleotide. So these are nothing but the components of nucleotide, or it is the component of nucleotide. So you can remember the structures as well. Next, we see about how Watson and Crick, these were the two scientists which understood that how these nucleotides are arranged into the genetic material, that is DNA. तो जो structure उन्होंने describe किया था, it was that the DNA is present as a double helix. यानी उसके पास दो unbranched polynucleotide strands हैं. These strands are going to be linked together by phosphodiester bonds and they will run anti-parallel. Anti-parallel run कर रहे हैं, मतलब what do they mean by it? So one strand will be having 5 and other will be having a 3 prime end. So 5 and 5 दोनों साथ साथ में नहीं रहेंगे, वो उल्टे बैठेंगे. फॉर एग्जाम्पल एक की मुंडी ऊपर है और दूसरे की नीचे है. This kind of a fashion is nothing but called as anti-parallel. That means एक जाएगा from 5 prime to 3 prime, other will go from 3 prime to 5 prime. That was the movement of the two nucleotide positioning. अब ये तो जो strands होते हैं, these strands are going to be coiling upon themselves onto a common axis. Common axis के ऊपर they will coil and that will be showing you a helical structure. अब जो helical structure का जो backbone होता है, जो main उसका जो जैसे हर structure के बीच में, हर building के बीच में pillar खड़े करते हो, that will be acting as the major backbone, major skeletal. So जैसे body में framework होता है, so of the bones, so वैसे ही डीएनए का फ्रेमवर्क कौन करता है, बोन वर्क कौन करता है? इट इज डन बाय द शुगर एंड द फॉस्फेट कंपोनेंट. शुगर एंड द फॉस्फेट कंपोनेंट बैकबोन बनाएंगे, दैट इज फॉर्म द रेलिंग, वेयरएज द स्टेप्स जो होंगे वो बेस पेयर स्टेप्स बनाएंगे. सो हाउ डू यू फॉर्म डीएनए का स्ट्रक्चर? कैसे बनाते हो डीएनए का स्ट्रक्चर? आप कुछ ऐसे बनाते हो. So this part that you draw is nothing but the staircase ka railing. This staircase ka railing that is in black is the backbone, whereas the part in red that is nothing but your base pairs. These base pairs are forming the staircase. This is spiral staircase. एक साइड जो होगी इसकी it will be 5. इसका स्ट्रैंड कैसे जा रहा है, उसका अपोजिट यहाँ पे 3 हो जाएगा, और फिर यहाँ पे you will have 3, here you will have 5. So like that you will see that it is anti-parallel passing of the DNA. अभी जो बेस पेयरिंग होती है वो भी स्पेसिफिक होती है. बेस पेयरिंग स्पेसिफिक है. व्हाट डू यू मीन बाय इट? वी आर सेइंग कि ऑलवेज एंड ऑलवेज adenine will pair with thymine, whereas cytosine will always pair with guanine. Why is it so? अभी A और G ये दो कौन होते हैं? प्यूरिन्स होते हैं. C और T कौन होते हैं? pyrimidines होते हैं. So always it is a rule that one purine will be combining with one pyrimidine. So can you see that logic over here as well? One purine is combining with one pyrimidine. Can you see? वैसे ही होगा. Why? Because it is complementary to each other. और ये जो complementary base pairing हो रही है, these complementary base pairing is going to be because of hydrogen bonds. So hydrogen bonds जो होते हैं, कितने hydrogen bonds add होते हैं between these base pairs? So hydrogen bonds A और T के बीच में 2 hydrogen bonds आएंगे, whereas in the case of C and G there will be 3 hydrogen bonds. So likewise base pairing specific होगा और दोनों के बीच में कैसा होगा? It will be because of hydrogen bonds. Next we see this point is about the same कि A and T will be by 2 hydrogen bonds and guanine and cytosine will be by 3 hydrogen bonds. Then next part we have to see कि 5 prime end जो होता है, जो 5 end होता है, this 5 end is going to be what part? It is going to be the 5th carbon. Why is it 5 prime? Because it is the 5th carbon of the pentose sugar is free, whereas the other end, that is 3 prime end, उसको बोला जाता है because at that position the 3rd carbon of the pentose sugar will be free. So that is the reason why it is called as 5 prime and the 3 prime end of the polypeptide. Next we have to see that each base pairing, हमने जो अभी ऐसा structure draw किया था, this structure was having a turn into it. So this turn is how much degrees? So once you are checking the degree, that degree between each base pair is going to be very specific. और वो specific जो होता है that is 36 degree turn होता है. At every base pair the strand will turn 36 degrees. That means after 10 base pairs there will be a complete helical strand, one full turn, that is 360 degrees, will have how many base pairs? 10 base pairs. So उसी के इसमें you can also identify the distance between the two base pairs. So these are stacked at a distance of 3.4 angstroms. So 3.4 है तो 1 turn का कितना distance हो जाएगा? 1 turn का distance is called as pitch of DNA. So that will be 34 angstrom. So this is referred to as 1 turn. Okay, यहाँ पे 5 आ गए, यहाँ पे 5 आ गए. This is entirely 34 angstrom. But this part, the smaller one part, this will be 3.4 angstrom. So that same understanding can be done by the figure adjacent to you. So एक होता है minor groove, this is your minor groove. एक होता है major groove. So minor groove ये छोटा वाला part हो गया. Major groove जो होता है, this is your major groove. This major groove is the longer part और जहाँ पे बहुत ज्यादा turn होता है, that is minor groove. 10 base pairs के बीच में distance कितना होगा? The distance is going to be 34 angstroms. And between first and second base pair कितना होगा? It is 3.4 angstroms. और the distance between the two polynucleotide chains वो कितना होगा? It will be 20 angstroms. हर बेस पेयर कितना होता है turn? It will be 36 degrees it will turn. and one complete turn will be 360 degree. That means 10 base pairs will be required. Next we see is the Chargaff rule. Chargaff was also a scientist who was showing his experiments in the support of the Watson and Crick. चारगाफ के रूल की वजह से ही वॉटसन एंड क्रिक को एक आइडिया मिला that can be helped to identify that which nucleotide is combining with what nucleotide. So amount of purines and pyrimidines are always equal. This was one statement that was given by Chargaff. कि जितने प्यूरिन्स होते हैं उतने ही pyrimidines होते हैं. So A and G content will be equal to C and T content. उसी के basis पे Watson and Crick were able to identify how the DNA is sequenced. Next, amount of adenine is always equal to that of thymine. अब जब adenine और thymine का quantity same होगा, that means these two are always coming together. When they are coming together, then only their quantity will remain same. फिर हमने क्या किया, A और T और G और C का रेशियो निकाला. So जब हमने इसका रेशियो निकाला, so it will vary from species to species, but it will be constant for a given species. For example, A और T का जो रेशियो है A plus T by G plus C, ये रेशियो डॉग्स के लिए समझो 0.7 है. For cat it might be 0.4. This is just an example. सारे dogs के लिए 0.7 ही रहेगा. It is same for the species, but it will be different for the different species. So that is also a very important point that was given by the Chargaff rule. Last एक point बोला गया था, that is the amount of deoxyribose sugar and phosphate are always in equal proportion. So considering these different categories, the Watson and Crick model for DNA was proposed. So अब in the case of Chargaff rule, उसने Chargaff ने क्या estimate किया था कि he was understanding DNA which is double stranded. But what about RNA? RNA जो होता है, the most common form of RNA is nothing but single stranded. So single stranded के case में there is no complementary base pairing. Complementary base pairing जो होती है, it is absent. So because of that, the 1:1 ratio is not accepted in the case of purines and pyrimidines. तो उस case में Chargaff rule for single stranded RNA, the Chargaff rule is not applicable. Chargaff rule is not applicable. The next we see is ribonucleic acid. हमने अभी तक स्टडी किया DNA in detail, but what about RNA? So RNA के 3 forms होते हैं. One is the messenger form, second is ribosomal form, third is transverse RNA. Messenger RNA और mRNA ये कहाँ बनता है? न्यूक्लियस में बनता है. And what does it do? It will help you in synthesis of protein. It will take the code from the nucleus and it will come into cytoplasm. साइटोप्लाज्म में जैसे ही mRNA बनेगा, the ribosomal mRNA will come, the ribosome will attach onto it. This is the largest RNA, and this will now take place in the protein synthesis. Protein synthesis में ribosomal RNA will be helping. Now transfer RNA, जब प्रोटीन बन रहा है, हमें प्रोटीन बनाने के लिए अमीनो एसिड्स चाहिए. सो अमीनो एसिड्स को साइटोप्लाज्म में बना बनाया वापस लेकर आने को एक जगह पे you need a transfer RNA. So this is the smallest type of RNA and it is about 10 to 15% of the total cellular RNA content. ये साइटोप्लाज्म में मिलता है and it will function collect the amino acids from the cytoplasm so that protein synthesis can take place. So nucleus से information carry करेगा for synthesis of protein. Cytoplasm में आएगा, so it will form a ribosomal RNA. It will form the proteins और proteins बनाने के लिए जो amino acids चाहिए that will be provided by the transfer RNA or the tRNA, which is smallest type of RNA. Largest type of RNA कौन सा है? Largest is ribosomal RNA, smallest is the tRNA. So that was about in the details for the nucleic acids. हमने DNA के बारे में देखा, RNA के बारे में देखा. Now that was the end to the chapter of biomolecules. It was a quick session discussing the details of the biomolecules chapter. हमने carbohydrates के बारे में देखा, amino acids के बारे में देखा. We saw about the proteins, about lipids and also about the nucleic acids. So that was about it. We will come back soon with one more amazing session with the different chapters regarding 11th and 12th biology. So if you like this video, then don't forget to like, share and subscribe and do hit the bell icon for new upcoming videos. Till then we meet next time. Thank you everyone. Bye bye.